Right, bring it on. Question 5, help me. Parts of some seeds. Well, the first thing to sort of notice is some things have got 1 in, some things have got 2 in. The bits are shaded are kind of similar to each other. Name A to D. So A is the outside bit. That should be alright. Quite a lot of people got that right. B is kind of storage of food. And sometimes with these things in C as well, where you might get storage of food. But the difference between B and C is that C, there's sometimes 2 of them, and so that might jog your memory. And D is, if I said baby plant, if you come up with a better word than that, that's what we're talking about. So that's where fertilisation is taking place, and that's our new plant. So you need all 4 of those for 2 marks, 3 of them for 1 mark. Similarities and differences. A little bit complicated. Broad bean similarity with castor oil bean. So what's similar there? So there's all sorts of similarities between all 3 of them, so you can't use those. But between these this seems to have 2 of these things, and that has 2 of them as well. A difference is this white area here. So the castor oil has got this white area, and you need the name of it. The broad bean hasn't. Maize. So similarity with the castor oil now. So similarity with the castor oil, they've both got this white area here. And a difference, well, the difference between 1 of these things and 2 of these things. So if you can get the names for those right, then you're okay. There's 1 mark going across. A little bit of blurb about an experiment. It detects dehydrogenase activity. So a bit like your methylene blue experiment that you did. So this one's going to go red, where we get hydrogen's being produced in respiration. So we're going to get some respiration in this seed, and where are we going to get respiration using up the food? Well, we're going to get that where the living thing is that's producing the enzymes and so on. So where the living part of it is, or when the living part meets the food source. Here we have an experiment that you've seen in the lab. It's in your lab book. So you've either done this or you know about it, you've seen a demo. Name the enzyme responsible for the clear zone. So we've got starch here, and we're in the clear zone. The starch has disappeared, so the starch has been broken down into maltose and then possibly into glucose. But what's the enzyme that breaks down starch? You've definitely met that, and you know about that. 1 word. What was the purpose of the boiled seed? Again, your experiment had the boiled seed. There's a word for this type of experiment. Or you could give a description of why it's there. It could be that just having something sat on this plate makes the starch disappear. It's to make sure it's something to do with the seed. Particularly, you could focus on why was it boiled. This last part's really complicated. This is how 2 enzyme systems depend on each other. So one enzyme produces something that helps the other enzyme, and then in turn the other enzyme gives us something that will enable the first enzyme to exist. So don't just give us a descriptions of what the enzymes do. how do they depend on each other? That's really difficult.